let's get started, guys. A lot of homeworks. See you guys working. Okay, so I'm done correcting your essay. I will post the grades today. I just want to go over all the comments I put there one last time. I adjusted your grade for assignment number 4, so you should have the right grade right now. So I gave a point to everyone who found dystrophin to not penalize people. What else? Assignment 3, the grade is also online, so that should be it. So it will give you about a week before you have to submit essay number 2. So in terms of essay number 2, two things that I kind of highlighted the points that most of the people got right and the ones that people should try to work on if you want to get a very good grade. So let me start down here and then I'll go back here, okay? So proofreading and references. So I think this is unrelated to this class. Honestly, your writing right now, unless this is your first year in English or whatever, should be almost perfect. But it is not the case in your assignments. Like the comma, there's a space before it, the references after the dot, the period, you leave mistakes, like Word puts the red thing, but for some reason you don't correct it. So I don't know what the hell happened. Maybe there was too much work and you were just like, structural genomics, let's just do it quickly, right? But technically, automatically, your writing should be like good from the beginning, right? If it's not, try to practice this. For now, for your grades purpose, find some sort of tool that can help you correct it, okay? But the mistakes I see in here is not 5th year or 4th year or 3rd year, even university level, okay? I'm not saying everyone, but some people kind of destroyed this thing in terms of grammar. So this is just for the grammar, right? So, and the writing style, right? Obviously, we're in science, so sentences tend to be like 3 or 4 lines long. But just for yourself, try to be more concise, one idea per sentence, so that like you get into the habit of it, right? So that then your writing just becomes automatic. You don't even need to think, oh, this sentence is big, I need to cut it into multiple pieces. Just get into that habit. The second thing, references. So these things you should have kind of practiced your whole life. Just put references everywhere. It's actually easier to just put references everywhere than not to put and think, should I put one here? Should I not put one here? Just put it everywhere. Take one article that's a review of the topic that you're talking about and put it everywhere, right? That's the easy trick. Obviously, in the assignment, you had to discuss, you had to relate this paper to other people's work. So this is where you go and find other papers. I'll just give you an example of how you can do this. KIP4A was discovered to be potentially a AR-independent prostate cancer specific driver. So then go find who else is working on KIP4A and what does it do in those, and then bring that to this paper, right? You can also talk about WDR62 and what other people are doing. You could be innovative and talk about the application of clinical and functional, this similar pipeline in other fields. So this was kind of open, but unfortunately, I think there's a lot of people that got it right, but you could a little bit improve on this part here by remembering that this is part of a bigger picture. And so that bigger picture means that there are other people that are working on it. So if you find KIP4A in this paper, which was in 2021, I think. I forgot which year this paper was. But now we're in 2024, right? So probably there's something about that you can use right now. So that's that. The other thing that was kind of not the best was the idea that you had to discuss conflicting data or alternative hypotheses. And I know this does not come easily. You have to kind of think of it a little bit at this level. But I'll give you just an example of conflicting data. We say that this pipeline kind of finds all the genes that are prostate cancer specific, right? But the only two that they're talking about that are already known is AR and HuxB13, but in reality, there's tens that are already discovered. How come they didn't mention that, right? So this is something that you could have been like, wait, yeah, they're saying this is supposed to give you the top hits, but literature says there's gene X, Y, and Z that are part of prostate cancer, but they only talk about two, right? This part is a little bit hard in here, but if you think a little bit creatively, you can kind of figure it out. Alternative hypotheses, what else? You know, KIF4A kind of seems to be cancer specific, but they only tested 6 cell lines. And so what about liver? What about, you know, the other organs? And as a matter of fact, just as an example, protein expression atlas. So, no, not this one, the human protein atlas. Okay, so you're not supposed to know this, but just to explain to you, because you need this kind of perspective when you're reading papers, right? So they're saying that this is prostate specific and causes cancer just in prostate, but if you go here, you have breast invasive carcinoma, which is kind of the top, right? So it's kind of weird, right? So this kind of discredits a lot of what they're saying. It's just at the time they had it there, right? And if you look online for the expression of this protein, for example, so the way you would go about the thinking so that you can start building this way of thinking is, okay, they have 2 proteins. They're claiming things about these proteins. Let me go search about these proteins. And then you go for KIF4A, and you look for its expression. So right on Google, KIF4A expression in other cancers. And you'll see that actually prostate cancer is not really there. Now, if you do AR, let's see, right? Even AR is in breast cancer, which is kind of weird, right? But we're discovering more and more that AR is actually involved a lot in breast cancer, okay? Especially for women who have polycystic ovary syndrome, PCOS, and many other, let's say, diseases that lead to this kind of cancer, okay? With that being said, obviously, if you take a look here and you remove the outlier, you put the outliers, then you're starting to kind of bump up the other stuff, but, you know, who cares about this, right? So this is a bit hard. This is a bit hard to do, but this is basically the actual part where you act like a scientist, okay? What you're very good at doing, because I guess you've been doing this for a long time, is talking about the background of the study, right? You talk about prostate cancer, functional genomics, clinical genomics, CRISPR-I, you talk about statistics, you even bring in some other studies related to prostate cancer, so this is good. Obviously, you're able to talk about the data very well. Most of the time, try to not jump from one result to the other. Instead, do kind of a transition between them. So what do I mean? We did X and we got A. Then we did Y and we got B. Because we got A, it means that this, so then we did Z and got, okay? And then you continue. Not like we found 1,472 proteins. We did some filtering and then we got A. 4 genes here and 4 genes there, right? So try to have kind of a logical chain there, but most of you got this right, and you're very good at future directions, always curing all the cancers possible, and the significance of the study, okay? So please, this time, try to have a better proofreading of your text. I know you have a lot of work to do, but there are tools online that can help you, and they'll have a perfect text, right? It will make it easier for me to read, because I still have to grade you on the content, even though the writing is like, there are a lot of mistakes and big sentences that are convoluted, okay? So, and the references, if you end up with like 3 or 4 references, start asking yourself questions like, what are you doing wrong, okay? Already just relating to other people's work, that's maybe like 2 or 3, and then you have at the top like 2 or 3 statistics, and then you're explaining a technique or 2 techniques, you're already at like 6, 7, 8, okay? With that being said, today's lecture, we're going to talk about gene therapy, I will not get in depth into this because there's a lot that we can do, but I think I'm going to start the lecture by talking about where all these genetic engineering started, okay? So Sanger just keeps getting freaking Nobel Prizes. This is his second Nobel Prize that he got, along with Paul Berg and Walter Gilbert, and they were the first to insert DNA from one organism into the other, okay? So this is where genetic engineering started. Paul Berg passed away last year. The others are already gone. So I put his picture here, and this is basically the paper that got him the Nobel Prize. All he did was, well, all he did at the time, it was a lot, but all he did was take genes from glycolysis and put them in the SV40 plasmid. And this is a Nobel Prize. This is a Nobel Prize. Obviously, he has a career, and he did a lot of work to get there, but at the time, this was almost impossible. It was very hard to do, right? And this, you can do it now in the lab in like maybe 2 hours. Okay, so this is where the field of genetic engineering started. Then from there, it went crazy, okay? The first thing that came up was the yeast knockout collection. So if you remember from one of the lectures, I told you that we have a knockout of each gene, and then there's a group in Toronto trying to build a double knockout. So they basically use genetic engineering, and the way they do this is through homologous recombination, because yeast is very good at homologous recombination. So they take a cassette here that has a kanamycin resistance gene, and then they basically add to it these adapters on each side that are homologous to the sequence of the gene they're trying to knock out, right? And so what happens is that when you put this cassette inside the cells, obviously you're going to make an up tag and a down tag for each of the 6,000 genes that the yeast has. Homologous recombination in yeast happens very well. It's a nice organism if you want to work on that. And all that will happen is that because they have similar sequences here, not complementary, but same sequences, they will kind of switch or recombine, and then you'll end up with your gene being disrupted, right? Your gene of interest. So then when you grow the cells on a plate and you add kanamycin, only those colonies that have the gene disrupted because of the insertion of this cassette will survive, and the other ones will die, okay? So this is kind of the basis of all the genetic engineering. You have some sort of homologous recombination, or you do something like this, where you're using nucleases to cut and paste, okay? From there, what happened, just a little bit of history starting from yeast, and then there's many groups that started kind of playing God and trying to create different organisms from scratch. They created a functional living yeast that had only a single chromosome, so they took pieces from each chromosome and then put them in a single chromosome, survived. They also created a synthetic yeast genome. You remember this paper I told you about? They removed like useless stuff in the yeast. About 8% of the yeast was removed. This still survives. There's a slide I removed because I didn't want to spend too much time on this, but I think I'm going to talk about it. They also created a yeast E. coli that had only 61 of the codons instead of 64. And I think they made even a simpler version where, because each codon codes, so one amino acid can be coded from multiple codons, so then you can remove the redundancy and still have a functional organism. And there's Craig Venter that did some crazy stuff in this, creating bacteria and yeast and whatnot, so you can look into that. We're more interested in humans. So the topic today is genetic engineering in humans. And before I get into it, can anyone tell me if we can do homologous recombination in humans? Like for genetic editing. Let's say you want to cure a disease. No? Maybe.
Can you rephrase the question?
Yeah. So if you want to remove a gene that's bad in the cell and replace it by another gene doing this.
Who can do it?
No, can we do this? Does that work for humans? Who said no? Yeah, what's up? Why?
Because the genome of the human is very massive and has a lot of sequences.
Yeah, so human genome is full of repetitive sequences. So what happens is that when you try to do this homologous recombination, you end up having off-sites. So you're going to basically do homologous recombination with other sites. The human genome is so big that for you to target the right spot, it's almost impossible. So you need something that is more direct, more guided. Obviously, the word guided, you know where I'm going. But that's basically the answer here. The other problem, so the initial strategy is actually to do this because you don't have CRISPR at the time, right? So you had to do it some sort of way. So people were adding these selectable markers or ways that you can detect the insertion at the right spot. And so because those homologous recombination events were super rare, okay, for you to get this, most of it will be actually useless. Some of it will actually look exactly how you want, but you can't do this for humans, right? You can't just start putting antibiotic and killing everything, right? So instead, you have to use some sort of selectable markers that you can see so that you know which ones actually integrated your genome and not, okay? But obviously, this does not work very well. If you're trying to cure some sort of disease, you'll probably also cause other problems, okay? We still do have a way of doing it for mammalian organisms. So in the labs, we used to do it like this. Now it changed a little bit. So the way it works is that if you're trying to disrupt a gene, so we didn't talk about replacing a gene yet. So here it's just disrupting a gene. So let's assume you have this gene here that has an exon 1 and 2. One way that you can disrupt this gene is by breaking exon 2, for example, right? So the way you do that is by inserting something inside of it. And by putting something inside of it, then you create you basically break the protein into two parts, right? And in this case here, you could insert, for example, the neomycin resistance gene. And that neomycin resistant gene will help you in selecting the cells that integrated the neomycin in it, okay? So you have your clone gene, you have your targeting vector. So now you're going to create your basically piece of DNA that you will put in the cells, okay? And you do that like this. So first of all, there are three options that can happen. You can have the vector that contains your neomycin gene, so basically this here, recombined with the chromosome of the cell that you're interested in, and that will confer this resistance to neomycin, right? Because there's a homologous recombination, you're going to leave this red part here, which is another selectable marker, okay? So then if you're looking for a chromosome that inserted through homologous recombination and doesn't have any other weird stuff, then you're going to select your cells using neomycin resistance and then this TK minus. You're not going to add the antibiotic that will kill the cells, okay? So let me backtrack. TK is thymidine kinase. It's an enzyme that is found in herpes simplex virus. And when you add to it a chemical, a drug, it will metabolize it and then turn that into something toxic and kill you. So you can use it or kill the cell. So then you can use it as a selectable marker along with this. So then let's look at this example here. If you have an insertion of your vector with the gene and all that, just random insertion, then you're going to bring with you the TK plus the neomycin, right? So then you have to add neomycin plus another drug, and because that drug will kill these cells here, you won't have them, right? So because this you're not interested in. You're interested in this here. In this case, because you don't have that TK enzyme, then you cannot metabolize one of the drugs that you give the cells, and so you cannot turn it into something toxic, so they survive. I know it's a little bit complicated here, but just the way you have to understand it is that these insertions can happen at different places. You want it to insert exactly where your gene is. So for that, it will use these parts here as the ends that will recombine with the vector, and then that will recombine with the chromosome because they have the same sequence. If they enter like this, you select like this to get them. If they enter randomly, you have to select like this to get these ones here. And then if there's no insertion, you have to select like this, okay? So just 3 different ways of selecting. That's all you need to understand here. So when you have your cells, some of them got this, some of them got this, some of them got this, then you have to select for them. So you're going to add neomycin, you're going to add ganciclovir, and you're going to just wait for it to basically kill all the cells, leaving only the ones that had this scenario here where there was a homologous recombination between your vector and the chromosome that contains your gene, okay? This is an old way of doing it. And then one step further, you have to put it into the, so these are embryonic stem cells, obviously. Then you have to put it into the blastocyte, if you remember from your mechanisms of development or first year biology course. Let it grow and hope that the cells that are produced here from this stem cell are your germline cells. Because what happens is that the first time mice will grow, you'll make a chimera. Some cells will have your modification, others will not. Your goal is to get the ones with the germline modification that has this, this insertion here. So then you can use the germ cells to make actually purebred mice. Okay? Make sense? And then you just keep mating them and doing some genotyping using some primers until you find the mouse that has that modification that you want. Anybody has a question about this? Like Haiti? No? Okay. Yeah, I know it's a bit complicated, but it's actually not the best technique. Right now we use CRISPR, so it's much more easier. You choose your DNA sequence, you put Cas9, and then you add the sequence that you want to be inserted, and that's it. It's just before we had to do some weird ways because we didn't have CRISPR, right? Okay, so if we have the technology to do it, how come we still don't have any? We didn't cure any disease yet with genetic editing. Yes, Gabrielle.
Part of it's like ethics, like we're not really allowed to do that.
Yeah, we're not really allowed to do that, but why not? Why are we not letting it happen? Do you guys know? Yeah, Akil.
Is it something along the lines of like the bounds of what we can define as like acceptable is a little bit gray, and then certain circumstances and certain contexts can be ethical, but in different, it could be non-ethical. It just gets really muddy, and rather just not touch it at all.
Yeah, so basically I'm going to re-say that in a different way. If we allow this to happen, to cure disease, how come we don't allow it to make a certain group of the population better than the others? Right, so eugenics. So this is some crazy stuff. There's a big debate on this stuff here. There's people that want it for just disease, but then the others are fighting, saying, okay, well, we want it for also making humans better. Things like this. So it's very complicated. There are many other reasons. Do you guys have any idea? Think about, yeah, Maria?
Well, if you're curing diseases with this genome editing, people that take medication regularly, things like that, they're not going to take that medication. So I guess
Oof, very good point.
to not use it so that we'll have pharmacies that,
We are basically talking about the pharmaceutical industry being being very illegal there. Yeah. 1 second, Akil. Sarah?
there's also biological challenges like DNA repair.
Yeah.
It's less effective and less active in mammalian cells.
Okay.
So we can mix like editing.
DNA repair mechanisms are less active in mammalian cells, so that when you break the DNA, you might cause any problems. Is that what you're saying? Yeah? I'm not sure. I didn't... is DNA repair less effective in mammalian cells? I have to think about this. This is a good point. Akil, then Julian.
If the technology becomes monopolized by private companies or certain countries or whatever, it could only be accessible to a certain, population. Certain socioeconomic class or whatever, and then that creates the basically like a whole eugenics issue.
Yeah. Yeah. Julian?
We might have the
Yeah.
located like this user, and it's nowhere near like finding loops and data and story. It's not like a computer at all. It's extremely messy. There's
Yeah.
change reactions everywhere and strange interactions, and we don't actually understand how this entire tangle works. So even if you can make a change, actually achieving the desired phenotype is extremely difficult.
Yep. Matthew.
If you're not modifying the germline cells, then it's really hard. Like when you're working in the lab, you just, you transform a population and then you just kill all the ones that didn't get it.
Exactly.
You can't do that if it's like with one person who you're trying to cure a specific thing.
Yeah. So this is why there's delivery systems that are being invented. Mark? Yeah.
So for the gene editing that's occurred in China,
Yeah.
I think one of the concerns was they don't know it could happen that you don't know the entire function of a specific gene.
Yeah.
Like, so there may be future unintended consequences to.
Yeah, downstream effects that you didn't calculate for. Okay, good. So you guys like this topic, huh? Okay, let me tell you something then. So today, tomato engineering, today or yesterday, there's a paper that came out. Obviously, you would expect us to be much further than this, but there's this team that actually re-engineered tomato to make it 20% or 30% sweeter. So this came out like 2 days ago. This seems like something very stupid to do, but actually tomato is a very complex genome. So if we were able to do it here, there's potential, right? That's the first thing. And obviously, they didn't do this just randomly like that. Sweetness is the main thing that gets people to buy tomato. And it's one of the most expensive fruits or vegetables, depending on how you see it, okay? So this came out just 2 days ago. Sounds very stupid. They make it bigger. They made it bigger than the normal tomato and 20 or 30% sweeter. And they're using these new technologies to edit this enzyme. So you can read the paper here. And then just go over it. We're not going to do that now. We have better stuff to do, okay? So we do have the technology to do it. It's just we're a bit scared of what could happen downstream. That's the first thing. The human genome is crazy complicated. It looks very simple, A, C, T, and G, but nope. Most of the time you break something in the cells and then you expect something and then you end up having the opposite effect of what you were waiting for. That's just because there's other stuff interacting with it. And you think that, for example, an oncogene sometimes will transform into a tumor suppressor, and you learned your whole life that that's an oncogene, but for some reason in your condition that you're working with, it's acting the opposite way, okay? So it's very hard to predict. The other thing is that we're scared of the politics and eugenics and people claiming things and, all that extra stuff, let's say. So current gene editing tools. There's a few of them. You probably heard about them. The zinc fingers, which are, they call them zinc fingers, but it's more like a protein, an endonuclease linked to zinc fingers that recognize DNA, and then the endonuclease breaks it. Talons or transcriptional activation, whatever. Forget about this, it's useless. Never heard of it being used, but there are the talons that also work like the zinc fingers. So they have this endonuclease linked to them. They recognize a different type of DNA structure, and then they just break it, okay? And then you have CRISPR-Cas9, obviously, which was developed by Jennifer Doudna and Emmanuel Charpentier. This is the real stuff, the real deal. Now, there's many variations of this that are coming. You learned coming out. You learned about CRISPR-I. You learned about CRISPR-A, and now there are others that target RNA. It's just going crazy. Okay. The idea behind these is just each one of them is better for something. CRISPR-Cas9 is the one that is most efficient. The others are less efficient because the way they bind to the DNA is kind of less stronger than this one here, let's say, okay? What's interesting about them here, though, is that they have less cytotoxic effects. CRISPR-Cas9 is a little bit more toxic and requires usually different ways of delivering the CRISPR-Cas9. You cannot use viruses for this because the Cas9 protein is big. And so you have to use different methods like liposomes, like nanoparticles, things like this. The other thing is that there are off-target effects with all of them. In this table, we're only talking about off-targets here, but off-targets here too. One thing that is not nice with CRISPR is the fact that it needs this sequence near the target site called the PAM sequence. So if you don't have it, then the CRISPR-Cas9 complex will not actually bind to your DNA, okay? But in general, this is the best, obviously, right? So let's talk about CRISPR. You already know what it is. It's short for clustered, regularly interspaced, short palindromic repeats. It was found in bacteria because it protects them from viruses. So what happens is that it's basically the first immunity that we are aware of. And bacteria, when they get rid of the virus, they kind of capture some sequence from it and include it in their DNA. And they have this Cas9 endonuclease, let's say, that can then bind to this sequence. And then whenever a virus comes back to infect the bacteria, then the bacteria can destroy its genetic material, okay? Most of the tools that are made from created based on CRISPR, so all the variations of CRISPR, they come from the CRISPR of S. pyogenes. You probably heard about this bacterium. The only difference is that what happens is that Charpentier and Doudna, they were able to switch the system in a way so that it only required one piece of DNA, of RNA, called the single guide RNA, because before it required two pieces. And so they kind of improved it a little bit, right? And the single guide RNA is a template for the part of the DNA which you want to target your complex. Now, there are a few rules when you're designing such a system, because you can now build your own CRISPR-Cas9 system. You're going to have usually the same Cas9 enzyme. Sometimes you'll have a Cas9 enzyme with different proteins attached to it. We'll take a look at that. But the idea is that you're going to use the single guide RNA to find your target sequence. And that also needs to be close to this protospacer adjacent motif. And so the way you design this, you need to design it close to this sequence on the DNA so that you're able to get this whole complex to bind here. If this is not present, this does not work. It works, but not good. So what happens if your gene does not have this PAM sequence? CRISPR-Cas9 does not apply to it, right? They're starting to have different variations that bind to different PAM sites, but typically your PAM site is any nucleotide, then guanine, okay? So this is it. Your single guide RNA is usually 20 bases, approximately. The idea here is really to just bring this whole complex here and then break the DNA if your Cas9 is active. If it's dead, like in the paper that you saw, it will not do anything. But then usually when you have a dead Cas9, you'll attach to it a protein here that will do something, okay? So that's for breaking the DNA. Now let's say you want to replace a gene. So there are ways to do this. Obviously the cell has already the repair mechanisms that you probably learned in all your classes. But if you're trying to use this system to basically replace a gene or insert a different copy of a gene, then you can first cut it. And then after cutting it, you can have your gene just your DNA coming back together and having that gene disrupted. Or you can add a sequence that you're interested in, which is called your donor DNA, and that sequence will do some sort of homologous recombination with the double-strand break here that you created with the CRISPR-Cas9 and insert whatever you want in there. For example, if you have a point mutation, you can use your donor DNA to just fix that. So you break that part and then you replace it by homologous recombination. Okay? And then there are many variations of this. So some of the modifications that were created for CRISPR-Cas9, you can inactivate the Cas9 and fuse it with a transcriptional activator. If you're interested into breast cancer, you'll probably add some sort of transcription factor like estrogen, progesterone receptor, something like this. If you're interested in inactive, yeah.
Can the single guide RNA be longer than 20 base pairs, or it should have like a specific size?
I am actually not sure, but I think usually when I design them, it's between 20 and 30, but I can actually Google this, we have internet.
If we can do longer single guide RNA, maybe we could.
No, you can actually do it even longer, but I have to verify this stuff here. It's a bit weird from 100.
You said it should be
I think that's
17
the
to
whole,
Yeah.
whole guide, right? The targeting part is usually shorter.
17 to 24.
Yeah. So, okay. So this piece here is about 100 to 150 from what I'm reading, but obviously I have to check this up. But the piece that kind of guides you to the site is about 20 bases. In general, in all that you're going to do in all biology, it's usually around 20. Okay. Your primers are 20. Everything is 20. Okay.
If we could do longer, maybe it would be possible to target more different sequences, like it would be more specific.
True. The problem is the Cas9 that you're using. Because the Cas9, so if you remember, we learned about all the functional RNAs. The structural features here are important so that the Cas9 kind of recognizes the single guide RNA and does not bind to mRNA, let's say, or something like that. So there's a size issue that happens here, right? But yeah, for sure. But usually, so in technically, if you have 20 bases and you count all the probabilities that you can make with all the 20 bases, it's very rare that you're going to have off-target effect, but it does happen because sometimes the A will bind to another A or whatever, okay? So it's not perfect, but it's coming up. What else? Sometimes you also like this effect because you want to target multiple genes that have kind of similar sequences, and the people do this. Like you use one Cas9, one single guide RNA, but target multiple genes of interest that have. Exact. What else? You can add a transcriptional repressor like CRAP. We saw this last lecture. There are many others that you can use. If you're doing just research in the lab for fun, you can add GFP. So let's say, for example, you want to select for your cells that have the insertion and all that, then you can use GFP, and there's a machine called a flow cytometer. The flow cytometer will basically take cells one by one in your plate, and if they detect GFP in the cell, it will sort it to the right, if it's or to the left, and if it's not there, it will sort it to the right, and then you have basically your two populations that are separate, and you know that these cells have your insert. Okay. There's a lot of things to do with this. Every day there's a new way of doing this stuff here, so it's becoming more and more common. As a matter of fact, it's so common that you can basically just go play with this by yourself. There's CRISPR Cas9 design. They have a tool. You can just make a plasmid that contains your single guide RNA and all that, and then just order it, and then all you have to do is follow the protocol, and boom, there you go, you're done. Okay, it's very simple. Actually, you don't even need to think about how you design it. You just put the gene, which organism, and it does it for you. Okay, so that's how simple this stuff here became. So let's say we want to cure disease now. Okay, there's a few things that you need to do. Sounds very simple, but actually it's a little bit more complex. You need your sequence for the single guide RNA, and you need to put that into a plasmid. The plasmid will also contain the Cas9, and then it will be expressed inside the cell. You'll need to put the Cas9 under a Pol2 promoter, so a general promoter, so that you have it all the time. And then even the single guide RNA, obviously. You don't want it to, well, let's say, for example, you want to do a direct target in liver cells. So every cell has a different set of transcription factors that are active in it, right? Estrogen receptor is usually active in, for example, breast cells or cervix cells, right? And so if you're trying to target just those cells, then potentially you'll put it under an estrogen receptor promoter, right? A promoter that responds to estrogen receptor. If you're trying to target all the cells in your body, then you'll use a Pol2 promoter, which is basically going to code for everything, okay? If you're trying to replace, so now you basically use this, you put it in the cell will take care of it. It will make the Cas9, it will make the single guide RNA, they will go together, and as the things move inside, they will find the right piece of DNA and they will bind to it. Now, if you want to do a repair and replace the sequence, then you also have to give a repair template along with that, and hope, and I'm saying hope because it doesn't happen all the time, that this double-stranded DNA that will repair whatever is broken in your genome binds exactly how you want it, okay? Just to give you an example of how we do this in the lab. So you see those 384 plate wells. So basically you are gonna do a reaction with all the cells that you have, and then you have to take one cell in each well, one cell. So you use the microscope, pick up the cell and put it in there, and then you let them grow so that your clones are coming all from the same genetic background, and then you have to sequence each one of them to make sure that you have your insert. It does not happen often. You have like 5 wells that work out of this whole thing. But now there's robots that do it, or PhD students, depends on which one your lab invests in, right? In any case, so it's possible to do it. Now I'm going to give you an example of this being done in a disease. Obviously, I keep talking about cystic fibrosis. I don't know why, but we're going to talk about that. Just a little graph here. Most of the diseases, genetic diseases, are caused actually by just a deletion or a single point mutation. Okay, seems like there's big changes that happen in there, but only 1 point mutation can destroy the whole gene. And I'll show you one example here for the cystic fibrosis gene. But before that, a little bit of sad stats. So do you guys know what's, like, you've seen people with this disease? When I was younger, I used to see people with those oxygen tanks in the bus, like just dragging them, and they had those respirators. Okay, well, that's cystic fibrosis basically, and your life expectancy is 29 years old. So that means me, I'm gone already. That's the first thing. And the second thing, you know, there's about 40,000 people, so it's a rare disease. Obviously, the United States are always the ones that have this data. There's about 40,000 children and adults living with cystic fibrosis in the US. So why the hell would you do a whole research program just for 40,000 people, right? Well, you know, each life counts, so that's not really the argument there. As a matter of fact, if you want to get a lot of funding, focus on these genetic diseases, okay? So very quickly about the disease, if you're not aware of it. So this disease, you have an issue with one of the channels in your body, and that leads to excessive mucus production. So mucus is produced in your lungs, it's produced in your intestine, it's produced pretty much everywhere. And so what happens with that is that if you have too much mucus in your lungs and you're trying to breathe, then you end up having tons of problems. So you end up with a lot of lung infections, coughing all the time as if you were kind of a smoker, wheezing, shortness of breath, even male infertility, because obviously you know, you need mucus to carry out the sperm. There are complications that come from this if not treated. They will come anyways, treated or not. It's just, you know, you're trying to make the life of these people a little bit better. So infections all the time because this stays full. Your body's not able to clear it. Inflammation, which leads to cancer, and later on respiratory failure. So this is a very bad disease. That's why they need respirators, because it helps them get the oxygen in a more concentrated form, okay? CFTR gene. So this is one of the longest genes in the human genome. It was cloned by these 3 people. The focus is Francis Collin, which is also the director of the Human Genome Project. And he also found some dystrophy, muscular dystrophy genes. Just crazy career, okay? The gene is about 250 kb, 27 exons, and is found on chromosome 7. And this whole thing here encodes a transmembrane protein that allows for chloride to pass. If chloride does not pass, what happens? You end up accumulating mucus outside because there's water, the whole osmosis story from 2 to 5. And then your cells, because you have too much chloride, you end up also producing bicarbonate and a lot of crazy problems, okay? Just remember, this leads to a lot of mucus, and then you lose your ability to breathe. So there are a few treatments that exist to fix this problem. So obviously, this gene is so big that you have multiple mutations that lead to it becoming dysfunctional and not allowing the chloride to pass through. There are a few drugs that are already being used, like ivacaftor. They all finish by Caftor. I don't know why. I Googled it, can't find anything about this. But I guess it's the WHO's determination of drugs that target specifically CFTR. So everything that finishes by Caftor, it's for this disease. So these channels, they have a gating mechanism. Sometimes these mutations will break the gating mechanism. So this enzyme, this small molecule here will actually potentiate the channel, allowing it to be kind of better at transporting the chloride. There are also others that improve the folding of the protein. So there's one mutation, one single mutation that breaks the protein completely. And if you put these small molecules, they'll improve the protein folding of the CFTR protein and bring it to the surface. So these are interesting, but they don't solve the problem forever, okay? You have to keep taking these drugs your whole life. People tried viral approaches, so you infect the lungs directly and you infect your whole body with some sort of CRISPR system that changes the gene inside. Because if you're able to switch that single point mutation that causes this problem, then you can fix it, right? Technically. Yeah? Sorry, if
you only have like one point mutation,
yeah.
would it make it easier to cure, or like it doesn't
Yeah, it makes it easier to cure as long as you're able to target properly the gene, right? Because CRISPR has been modified to do these base repairs, so a single base repair, and you can do it, and this is actually the technology that's being used for this work here. So the bigger the sequence you have to play with, the easier it is to fix. The problem is not into how you're fixing it, it's more into getting it only there, nowhere else in the genome. Right? That's the problem that is faced by these people that are doing work on this. Previously, what they were trying to do is, like asthma, you use these nebulizers, those sprays. So you can pretty much nebulize anything, right? You can nebulize plasmids, you can nebulize viruses, you can nebulize whatever, and just send it through. Goes in, infects your cells, and then puts in the plasmid of interest, and then your cells take care of the rest. The problem is that when you're using viral approaches, immune response. So if you need multiple times, then every time that you're taking the thing, you get an immune response, and your body just keeps reacting to it. And also, the luminal surface of your lungs does not really agree with viruses. It's actually meant there to prevent viruses from infecting you. It's one of your primary defense mechanisms. So this was abandoned very quickly. Right? The third option, which is the one that is being used right now, is, and obviously the Cas9 and all those stuff, they don't really fit in the virus, so you had to use different types of viruses and try to make them the least possible, make your immune react the least possible to this. So obviously, if you're doing a clinical study on this, you give them the virus, they get an immune response, they come back for a second dose, third dose, and they just keep getting these. So it's not very practical for a clinical trial, because right there you fail phase 1, which is trying to make it not toxic. So phase 1 is where you're trying to make sure that the drug is not toxic. Okay? Non-viral vectors, these ones are the best. Nanoparticles, gold particles, whatever particles you heard about, the best ones is probably the liposomes. So the liposomes, you put your DNA in them. So we have a few examples of this in the labs, like lipofectamine, which you probably heard about. You put your plasmid in there, inside the liposome, and then nebulize that, goes into the lungs, and then from there it's just history, let's say, okay? So I took this paper from the Lancet, one of the big journals there, in medicine. They did a clinical study using non-viral gene therapy for these people, and they had some pretty good data, or at least, I mean, they didn't cure it, but they stabilize the patients because these guys, they just go basically from being able to breathe to 29 years old, not able to breathe. And then obviously it's lethal. So if you're able to stabilize it, you still have the disease, but at least these people live a better life, let's say, okay? This is the first non-viral gene therapy that was done for this disease. So this is kind of a very important study. I told you that these lectures will be kind of examples of what you could do for your papers. Today I'm talking about the methods a lot. That's just because it's a clinical study, and so you have to focus a lot on the methods. So you can kind of think of it this way, right? So the way they designed their study, first of all, it was randomized. I will tell you what that means in a second. Double-blind means no one knows who's getting the treatment and who's getting the placebo. Controlled, and then phase 2b trial, just forget about that. But basically, they confirmed that it's not toxic, and now they can increase the dose and make sure that it's actually doing the effect that they needed, okay? They took participants that are older than 12 years old and had, so these are all the criteria they had for their filtering, let's say. They had this forced expiratory volume. So when you go to see your, how do you say pneumolog in English? Person that takes care of your lungs, the doctor that specializes in taking care of your lungs. There's a test that they do where they make you breathe into kind of this tube forcefully, and the volume that comes out in 1 second, so FEV1 means in 1 second, that's the volume that will tell you if you're kind of healthy, your lungs are healthy, smoker, got this disease, that disease, or whatever, okay? So they took people that are between 50 to 90%, okay? So these are just their cutoffs. They didn't care about the mutations, so they mixed up the whole population. They had 2 sites where they were doing this study. Obviously, it got approved by this committee, and the patients, the parents were doing the consent for the children, and the adults were doing the consent for themselves, okay? Randomization and masking. So no one knew what the hell, who got the placebo and who didn't get the placebo. It was almost 1-to-1 for the treatment compared to the placebo. So they gave approximately the same amount of people. Half of the cohort was given the treatment and half of it was the placebo. Stratified by FEV just means that they grouped them by kind of subdivision of this here. So 50 to 60, 60 to 70, etc. By age, and then they had some sub-studies in here, okay? And dosing sites, so this way you have kind of an even data set. And you'll see when I'll show you the demographics and the people that were treated, you'll see why this is important. Not like that BS study where the CRISPR screen was based on, okay? Which was purely skewed towards one side, okay? Masking, so the participants and the investigators did not know who had what. Only the people that were given the drugs were aware of that. The reason for that is that if you're an investigator and I tell you this one has a drug, you start seeing success, okay? So you have to kind of hide it from the investigators as well. So the way they went about this, very simple. 12 months, every few days, they give them a dose of the treatment, and the treatment is very simple. It contains a plasmid of DNA. Obviously, you're not going to know what it is, but I can tell you what it is. It's a CRISPR system for sure that has the guide RNA, and then you have your lipid formulation. That's the only, since it is something recent, I don't see anything else that could be used here other than that, unless they have some sort of technology that we don't know about, but I would be surprised. And they gave them 5 milliliter of this mixture here every 28 days, plus or minus 5 days, because obviously you expect people to come back to the center and take the drug. Some of them are busy on this day, hangover on the other, so you have to account for that, okay? The control had 0.9% saline. So this, I can tell you right now, is a limiting factor. This is where a place where I would be like, that's not good, because the control is not a mixture of random DNA along with lipid. It's 0.9% saline, and I'm pretty sure that saline affects osmosis in the cells, okay? So weird stuff. In their paper, they say that this had no effect, but how did they check for it? No clue. Or actually in the clinical study. Technically, what you would have as a right control for this is a DNA plasmid that is just scrambled, that does not exist anywhere and does not do anything, as long as it's a piece of DNA of about the same size with the same amount of it and the same amount of lipid. So this here, if I was going to talk about this, this is one of the issues I see. Their goal was just to see how your FEV evolved. Do you get better at breathing? Your volume, does it increase or decrease with time? And compared to the control. But they had also sub-studies. So they looked at, they also performed their bronchoscopy, and this is just looking inside your lungs to see how your lungs are doing with different factors that we'll show you now. They had other sub-studies. So their primary endpoint was to figure out the FEV, so the change in FEV. If the volume increased with the treatment, that means you're a bit better able to breathe, so that means probably there's less mucus, but you still do the bronchoscopy to make sure that there's no mucus, right? The bronchoscopy, they used a few things. They looked at lung function. They did CT scans. You probably know what CT scans are. If you go to the doctor and you have some sort of problem, they will most probably do CT scan to see what you have. They looked at other different endpoints. So these are called clinical study endpoints. You will hear this a lot. You probably know about this. They also looked at exercise, activity, inflammation markers, the amount of DNA in the lungs to see if actually the vector was taken in or not, the fitness of the CFTR transmembrane protein using electrophysiology, so put things, produce and measure electricity, for example. And so the main point is their ability to breathe better or not, but they had other endpoints because obviously a study of like $30–40 million, so you want to measure other stuff. This is what it looks like. So this is the pipeline they had. They started with 191 patients. They filtered them. 4 of them were clinically unstable, so you don't want to test on them. 3 had liver disease, which has a link with this. I forgot what it is. One of them, I think, tried to lie to make the money out of this because when they evaluated him, he didn't have cystic fibrosis, so he got kicked out. After that, there were 151. 9 of them quit. 2 of them developed exclusion criteria, so obviously you have to take them out. These studies are huge. You have doctors, pharmacists, biologists. Like, it's crazy, okay? And then the 140 are randomized, so you don't know who's who, divided into 2 groups, and then given, placebo or the mixture, okay? And then obviously as it goes, people keep quitting, and so they ended up with 54 that had placebo and 62 that received the treatment. Not bad. Okay, so demographics. The cohort in this case had looks like this. I have a question for you now that I told you about the limitations in the past one. Do you see any limitations here? No? Yeah, Maria?
Yes, the ones that are less than 18 years old or less than those that are adults.
Yeah. As a matter of fact, this is one of the most important factors. I told you that the life expectancy is 29 years old, right? So you want this group to be bigger than the other. Obviously, this one is important too, but some of them actually live longer than that, with treatment now. So this here probably I didn't look at the specifics, but probably there are some 50, 60-year-old people. Yes, sir.
Would the environment they live in affect it? Maybe someone lives with another person who smokes, or you cannot control the environment.
Yeah, there are some stuff that you cannot control, and people lie in these studies because they want to be part of it to get a treatment. Some of them, there's some stuff you can detect because they do a lot of tests, but others not. Actually, these were the first that were at danger during COVID, okay? Right, so this is one thing. That you get it, that's it. So these people actually have to avoid others to not get infected by anything, because any problem that hits your respiratory system, you're in really bad shape. Imagine already when you're like dying, from cold, and then on top of that, you're not able to breathe, right? Nose blocked, lungs blocked, the whole thing is blocked. So this is a very dangerous disease. The person itself, I don't think they smoke. But if your husband, wife, kid smokes next to you, then it makes it worse. But usually when you see the people like not in a good shape, you don't really smoke next to them unless you don't care. Yeah. Yeah. There's you're gonna laugh, but there's food that inflames your lungs also. So you have to, this is a horrible disease. You have to be careful from your environment. You see those pictures of smog, and some days it's very polluted outside, and the air index. So this affects them also a lot. So the environment does do a lot of things to them, and so these things you cannot really control for them. That's why you try to have the biggest cohort possible, right? In this case, the group of interest is mostly these ones, but because they're younger, usually the parents don't like to put them in the studies unless the doctor proposes it to you and you feel like your kid really needs it, so you just put them there. Yeah.
I had a question. It's not exactly about this slide, but so a treatment like this, is it limited to one generation of cells? If they need to reinfect again once that sort of
you keep taking the
size off, they have to take it again?
you keep taking it's like asthma. That's a very good question. You just keep nebulizing the whole thing, and it goes in your lungs. You spend a few days good, and then you take it again. And yeah, that's a very good question. Matthew, Alex had a question. That's it. Okay. In terms of the sex of the study, both of them are in equal numbers, so this is pretty good. They try to keep both groups close to each other, so this is very nice. What about the mutations? So remember I told you about the mutation that breaks the protein completely and is treated with one of the caftors there? Well, this is the mutation. So just one phenylalanine at residue 508, the deletion there just destroys the protein. It does not fold properly, and the membrane protein, and it does not shuttle into the membrane. Okay. Do you see any limitations here, or it seems to be pretty good? Yes, so I'm going to take Alex this time because you guys are...
It's clearly not ideal because you want every variety to be equally represented, but this is also going to be a reflection of the prevalence of the genetic makeup of people.
Exactly. Very good point. So there's something I didn't tell you about this slide. This is the most present mutation. So obviously your cohort is going to have this mutation, right? So this is representative of the genetic pool of society, okay? So this is pretty good. Maybe you expect a little bit more here, but you know what? If you take the population somewhat equally and randomize it, you'll end up representing the genetic pool, and so you don't really have to think about this. And what's weird about this study, though, is that I'm not doubting everything, but they say that they randomize it. For some reason, they always end up with approximately the same amount on each side, right? Now, the placebo group had 54. This one had 62. There's 8 different, so seems a little bit weird there. But let's look at the data. Okay, so all this 40 million for this graph here. This graph here. So this graph here, what they're looking at is the placebo, which is the saline. Every time they come, they measure their ability to breathe using this FEV, forced expiratory volume, and then they give them another dose. They let them go home, and then, as you can see, the placebo with time, they just keep degrading, and then they lose more and more their ability to breathe. This seems to be an outlier or something like this. 6 June, maybe they forgot. It was summer, so they were happy, or some stuff like this. The drug, though, the treatment, not the drug, the gene therapy, you could see that it was pretty stable. They stayed along the line of 0, so there was pretty much no change through time, approximately. So this is actually something that's very good because, you're basically telling them that they can keep their ability to breathe at a constant level and not have it degrade like this until they reach basically the point of lethality. Okay. They also compared this data here to other studies, and they found that is corresponds to basically a survival of 29 years of age. If you take each patient and you expand those here, instead of having them as averages, you'll have this like this, right? So the experimental group is looking like this, and each patient had a varied response, right? So in this case here, there's less participants, so there's this kind of gap here in the placebo group, but you can see that most of them are actually losing their ability to breathe. Okay. Forest plot. Did you guys ever see this? So this is the other stuff that they did on the side. So they basically compared the two groups, and they standardized the treatment effect using some sort of statistical tests. You can get into the details of this if, for example, you're presenting and you want to explain it quickly. But basically, you have the middle line here. If you see the bar moving towards this side, that means it's the treatment, it's favored on this group, and if it's on the other side, it's favored in this group. So if you take a look at their primary endpoint for the whole cohort, 54 to 60, so 60, so that means two of them were, I don't know what happened here, but you know, these points here is basically telling you the placebo and the treatment group, but sometimes there's patients that miss. That's just because they were not measured that day, or missing data point or something like this, or they removed it from the data set or whatever, okay? FEV, so you can see that the FEV is actually better in the side of the treated group. If you take a look at the safety here, for example, C-reactive protein, this is a marker that you'll get to test if you go see your doctor for inflammation, and just for immune system being overreactive. So CRP is a common test, so you can see that it's it's better into it's better in this cohort here, which have the treatment. If you were to use viral, systems, then you'll have this go back here. The reason why these guys have more inflammation, let's say, it's because this disease itself leads to a complication of inflammation, right? If you have mucus that you cannot expel from your body, trust me that you'll have inflammation all the time. So this kind of decreases the inflammation, most probably because it improves the lung function and all these metrics for the lungs fitness, like wall thickness. You know, these I'm not very aware of, but I understand that they're just there for the fitness of the lung clearance index. It even improves it also. But if you look at the p-values here, there is a change, but some of them are not significant, so be careful with that. This one here clearly significant. This is significant. So this is their main result. And you can talk about this in any way that is interesting. These two here, I don't really believe them in the test because they're kind of qualitative, and people honestly lie about this, or they have a disproportionate understanding of their own physical functioning and diet and all those stuff. So these would go here, and people think that they're like this, but actually they're not like that. So these are qualitative stuff that usually come from a questionnaire. There's a, what do you call it, an asterisk. You don't even need to read it. You can go read it, and you'll see that this is probably what I'm saying. I didn't even read it, but I'm pretty sure it's this, okay? These are actual measures that are done by doctors or biologists or things like this. Okay, one last thing. They looked at a lot of, obviously you have doctors in there and nurses, so they looked at a lot of symptoms to see if this treatment actually caused problems in the health of the patients throughout the time. This is called pharmacovigilance. You have to do this in phase 1. This is phase 1. You focus on this on phase 1 clinical studies, but you also do it in 2 and 3. And so you see that, so this is a 1.2. I don't know why they formatted it like this. I just screenshotted this. But you can see that they have approximately the same amount. I don't know what the Oh yeah, it's a number, it's the average time that they mentioned this symptom specifically from the whole cohort. So you add all of, let's say patient 1 said it 10 times, complained about this 10 times and the other 12, and then you just do the average for all of them, you see that they're pretty close to each other. So this seems to be quite good, not affecting a lot the patient's health and seems to be stabilizing their ability to breathe. Now, a few limitations in this study. The first one is the fact that they have no clue what it does in 10 years. So obviously, if you're talking about clinical studies, you have to talk about the future. You keep taking this nebulizer all the time, and this is a gene therapy. What are the chances that maybe one day it's going to do some sort of off-target effect and cause a cancer in there? So works, but scary. Scary works. In this paper, it seems to be perfect. I also talked about the saline control, which was weird. Do you see any other issues? No? Yeah. So, yeah.
They didn't, like, I know they didn't say explicitly what their treatment, like what was in their plasma, but they didn't evaluate like how much it went in, like how effective it actually was at integrating.
Actually, yeah, they did. They did. So that was one of their mechanistic endpoints. So they did measure the DNA amount, and they measured it only before, obviously, the basal, and then 24 hours after the 12 months. So at the end, they measured it.
The fact that like people didn't recover the full normal, like they sort of maintained their regular disease, I guess. So that was just because the efficiency of replacing that gene was not very high despite the treatment, or?
Exactly. So everything is a balance between fixing and breaking, right? And so in this kind of treatment here, it seems like those two are kind of equal. In any biological system, you have repair mechanisms and you have mechanisms that cause damage. The moment that the mechanisms that cause damage become stronger, then the cells just basically die. So in this case, it seems like they got it to a balance, but obviously probably they can improve it, right? And if they improve it, then the repair mechanisms, which is their gene therapy, will be stronger, and you'll see this graph here technically, where is it, go up, obviously to a certain maximum, because you can't, breathe 10 liters of air in one go. Okay? Make sense? That's it for today.